This is cannabis and it is awesome. Cannabis has been used to make fabric in China for over 3,000 years. It can also be used to make rope, paper, and even an alternative to concrete. We can extract oil from the seed for cooking and cosmetics, and some of the chemical compounds produced by cannabis have applications in medicine. But cannabis has a checkered history. Some cannabis plants produce an intoxicating compound called THC. Because of this, cannabis has been heavily regulated, which means that it's relatively understudied considering its wide range of applications. While it has been recently legalised in some parts of the world, there's still a lot that we don't know about cannabis, and this is particularly true with respect to its DNA. It's in the DNA of a plant, or its genome, where we find the instructions for how the plant should look, grow, and live. Most of these instructions are encoded in regions of the plant's genome called genes, and different genes have different purposes. But not every plant of the same species has exactly the same set of genes, and that's what my research is about. To shed some light on the genetic diversity of cannabis, I'm building a cannabis pan-genome. A pan-genome is a way of looking at the gene content of an entire species at once. It focuses on identifying the core genes, which are genes that are present in all members of the species, and the accessory genes, which are genes that are present in only some members of the species. The core genes tend to be the genes that are necessary for the basic survival of the plant, while the accessory genes are associated with variations in lifestyle. The type of pangenome I'm building is particularly well suited to finding out which physical traits are associated with which genes, and this has a huge amount of value. By knowing which genes cause a specific trait, we can then breed plants with combinations of desirable traits. For example, if we were able to identify a gene that improves disease resistance in cannabis, we could then introduce that gene into a fibre cropping plant, which could reduce the requirement for chemical use on the crop, potentially increase crop yield, and overall reduce the total cost to grow the crop. While this might sound like fiction, it's entirely possible, and a pangenome is a great place to start. Cannabis is awesome. We already know that. With a massive variety of applications, it's certainly not a one-hit wonder. I don't know exactly what a cannabis pangenome will uncover, but I'm really looking forward to finding out.